Hello friends, today we are going to study about the structure of ovule. This is an anatropous ovule. By anatropous ovule, we mean that it is a typical ovule, means that is commonly found and commonly studied. So, now we come to the structure of ovule. At first, when we see the structure of ovule, we see here an opening, and this opening is micropyle. Micropyle is the opening, or it is also said to be the mouth. By this opening, the pollen tube enters into the ovule. So, this is a way to enter the ovule. Next comes the funicle. Now, what is funicle? Funicle is a stalk by which ovule is attached to placenta. Now we see that there are some layers here. The first layer, outermost layer, is outer integument. By saying integument, we means it is a covering, and internal to it there is inner integument. So we see here that there are two types of integuments here. One is outer and the inner, and the other is inner. Both of these integuments are made up of soft tissue. They are made up of soft tissue, and they are made up of by sarcotesta. Sarcotesta. In the middle of these integuments, one more integument is found there, middle integument. Okay, so middle integument lies in between outer and inner integuments. This middle integument is a little bit different from these inner and outer integuments, as it is hard and made up of sclerotesta. It is made up of sclerotesta. Now, here we see that at one end there is micropyle. This is micropyle, and just opposite to this end, there is chaliceal end. There is chaliceal end. So, chaliceal end is found opposite to micropylar end. This part is called as chaliza. It is the place of origin of integuments. So by this we mean that the integuments are originated from this part. Okay, now we come to the Ambrose sac, which is the main part of ovule. So Ambrose sac is this part. It is in between the ovule. Ambrose sac is the female gametophyte. Is the female gametophyte. And outer the Ambrose sac, there comes nucellus. Nucellus is made up of parenchymatous tissue. Made up of parenchymatous tissue, and it gives nutrition. Gives nutrition to the Ambrose sac. Now, the end or the part where funicle is attached to the ovule, this part is called as hilum. This part is called as hilum. Now, if you see that the structure of ovule, which is anatropous or which we can call typical ovule, it consists of at one end the micropyle or micropylar opening. So, why micropyle is needed? It is required for the entry of pollen tube into the ovule. The outermost layer is called as outer integument, which is made up of soft tissue, that is sarcodesta, and inner to it is middle integument, which is different to outer integument and hard in nature, made up of sclerodesta. Now, the internal integument, which is the inner one, it is also soft and it is also made up of sclerodesta. Just opposite to the chalizal end, just opposite to the micropylar end, there is chalizal end, and this part is called as chalaza. So, chalaza is the place of origin of integuments. Here, in the center, we see that there is the Ambrose sac, the main part of ovule, which is called as female gametophyte. Now, this female gametophyte is nourished by nucellus, which is made up of parenchymatous tissue, and it gives nutrition to the Ambrose sac. Ovule is attached to placenta with a stalk that is called as funicle, and the attachment place of funicle and ovule is called as hilum. So here we see that the structure of ovule is a simple one and consists of all these parts. Thank you.